Hello everyone. Today we will know about the tertiary and the quaternary structure of the protein. In the previous slide, we have studied about the primary structure and the secondary structure of the protein. So what is the primary structure of the protein? So here the amino acids are present and these amino acids are joined together to form the primary structure of the protein. And now this primary structure of the protein, they are again coiled together to form the secondary structure of the protein. For example, this is the alpha helix. So it can coil together to form the alpha helix, or it can form the beta sheet or beta pleated sheet. So these are the secondary structure of the protein. So today we will know about the tertiary structure of the protein. So now this secondary structure of the protein, they are again coiled together to form a specific shape of the protein in the three-dimensional shape or the three-dimensional conformation. So the specific shape of the protein, it is responsible for the activation of the protein. For example, in the tertiary structure of the protein is called as the native protein. So what is the native protein? It is a functional protein. So that means when the protein is present in the tertiary condition, then only it is the functional. That means if it is present at the primary structure, in the secondary structure, then the same amino acid sequence is present, but still it is present in the inactive condition. So it activates only when it is present at the tertiary structure of the protein. So this is the native protein or the functional protein. For example, you can say, for example, the pepsin. The pepsin is the enzyme of the protein. It is present at the stomach. So the main function is to hydrolyze the protein. So now if the pepsin is present only in the tertiary structure, then only it is called as the native protein or the functional. That means when the pepsin is present at the tertiary structure, then only it can digest the protein. If the pepsin is present at the primary structure or the secondary structure, then the activity loses. That means in the primary structure and the secondary structure, it cannot digest the protein. It digests the protein only when it is present at the tertiary structure of the protein. So that's why this specific shape in the three-dimensional conformation is important, and this shape is maintained by 5 bonds or the 5 interactions. First one is called as the hydrophobic interaction. Second one is called as the electrostatic interaction. Third one is hydrogen bond. Fourth one, van der Waals force of the interaction. 5 one is the covalent bond or the disulfide bond. So the first 4 bonds, they are called as the non-covalent bond. So this is called as the covalent bond or the disulfide bond. So first come to the hydrophobic interaction. Let's imagine in the bucket the water is present and the water is the polar, that means it is the hydrophilic molecule. So if we add some hydrophobic molecules, let's say they are the hydrophobic molecule. For example, let's say soap. If we add the soap in the water, then they cluster together. They cluster together in the water molecule or any hydrophilic molecule. Why? Because this hydrophilic molecule, it cannot interact with the hydrophobic molecule. Because this hydrophilic molecule, it can interact with the molecule by forming the hydrogen bond or H bond. And hydrogen bond cannot be formed by the hydrophobic molecules. That's why the hydrophilic molecule, they try to minimize the interaction of the hydrophilic molecules. That's why they cluster together so that there can be minimized interaction of the hydrophilic molecules. Means now the hydrophilic molecule, they can bind only to this outer area only, so that there is minimal interaction of the hydrophilic molecules. So whenever the hydrophilic molecules, they are exposed to the hydrophilic molecules, then they attract each other to minimal the interaction of the hydrophilic molecule, and that is called as the hydrophobic interaction. Second one is the electrostatic interaction, or it is called as the ionic bond. So it is simply the bond is formed by the cation and the anion. So here let's say this is the NH3, this is the positive of the cation, or this is the CO minus. So here the bond is formed, here it is called as the salt bridge. So normally the salt bridge, the ionic bond or the electrostatic interaction, it is called as the salt bridge, but actually the salt bridge is called as the electrostatic interaction plus hydrogen bond, they are called as the salt bridge. But maximum people, they say the salt bridge as the ionic interaction or the electrostatic interaction. So here the hydrophobic bond is formed inside the body or inside the body of the protein, because at the outside the hydrophilic molecules are present and it cannot interact with these hydrophobic molecules. That's why always the hydrophobic interaction can only form inside the protein. So here also this is the electrostatic interaction. Here it is the amino group, it is the carboxylic group, it is the positive, it is the negative. So positive, negative, here the bond is formed, it is called as the electrostatic interaction. So third one is the hydrogen bond. Simply the hydrogen bond is formed by a del plus and del minus or it is called as the partial positive and it is called as the partial negative, or it is called as the, or what is the partial negative? The molecule which has a lone pair, it is called as the partial negative, or it is called as the electronegative. That means a lone pair is present. So because of this lone pair, it attracts the electron by this lone pair, and here the bond is formed, and the bond is called as the H bond or the hydrogen bond. So the hydrogen bond, most importantly, it is strongest bond out of all the non-covalent bond. So it is the strongest bond out of all 4 non-covalent bond. So fifth one is the covalent bond or the disulfide bond. Look at the structure. This is the primary structure of the protein. So here the amino acid is methionine, this is the valine, this is the cysteine. This is the structure of the cysteine. So then serine, glycine, then again cysteine, then the cysteine, then again cysteine, then cysteine, and again cysteine. So when the protein folding is occurred, then only the shape is changed to form the tertiary structure, and here the bond is called as the disulfide. Disulfide means disulfur group is present. So in the cysteine structure, it is the amino acid, here it is the amino group, it is the carboxylic group, it is the hydrogen group, it is the R group, and in the R group, here it is the sulfur is present, and this sulfur is responsible for the formation of the disulfide bond. For example, in this cysteine, here S is found, and here in the cysteine, here also SH, here also is SH group is found. So now the double S or the SS or the disulfide bond is formed and H2 molecules are released. So by that, here the disulfide bond is formed, and this is called as the protein folding. That means it creates a specific shape or the 3-dimensional shape of the protein, and this disulfide bond or the covalent bond is very, very important for the 3D conformation or the native structure of the protein. So why it is called as the covalent bond or the disulfide bond is called as the covalent bond? The covalent bond, it's simply the bond which is joined very tightly. So because it is joined very tightly, it requires much more energy to break down. So that's why the energy required for the breakdown of the covalent bond is very, very high, but in these 4 cases they are called as the weak interaction or the non-covalent bond. So means they require very low amount of the energy or the heat to break down. So out of these 4 non-covalent bond, the hydrogen bond, it is the strongest one. So this is the disulfide bond is formed. Here it is the disulfide bond is formed. Here it is the hydrophilic interaction is present. So these are the very different 5 bonds are formed, and these 5 bonds are always required to form this tertiary structure. So when only the protein is present at the tertiary condition, then only it can become the native or the functional one. Before that it is inactive. So always important that okay, well, I will say in the next slide. So this is the tertiary structure. Now it is, let's go to the quaternary structure of the protein. So this is the primary structure of the protein. So these are the secondary structure of the protein. That means either alpha helix or the beta pleated sheet. So this is the tertiary structure of the protein. So quaternary structure of the protein, it has a very different structure. That means in the tertiary structure of the protein, same protein, let's say the same protein, it is coiled together to form the alpha helix or the beta stranded. Then again they join together, they coiled together to form a 3D conformational or the 3D shape, that is the native protein. That is called as the tertiary structure. But the quaternary structure means here the more than one polypeptide chain is present. That means in the tertiary condition, here one single polypeptide chain. This is the polypeptide chain. That means the amino acids are present. That is called the polypeptide or simply called as the protein. So one polypeptide chain is present. But in the quaternary structure, here more than one polypeptide chain is present. For example, one most used example is the hemoglobin. In the hemoglobin, 4 polypeptide chains are present. 1, 2, 3, and the 4. The first one is called as the alpha, another is the beta. So this is called as the alpha 1, alpha 2. This is called as the beta 1 and beta 2. So there are the 4 polypeptides are present, and they are again joined together to form the quaternary structure of the protein. So what is the difference between the tertiary and the quaternary structure? Same 3D conformation is found, but in the tertiary condition, the single polypeptide chain is present, while in the quaternary structure more than one polypeptide chain is present. That's why the quaternary structure is also called as the multimeric protein, because more than one polypeptide chain is used. Another example of the quaternary structure is a DNA polymerase. Another is ion channel, and antibody. They are also multimeric proteins. That means they also have more than one polypeptide chains. So these are the examples of the quaternary structure of the protein. So now come to again to the tertiary and the quaternary structure. So here in the tertiary, the quaternary structure, a 3D conformation or the specific shape is present. So it is called as by the coiling of the secondary and the tertiary structure of the protein. But when the protein is subjected to change in the, let's say, pH, temperature, and let's say some exposure to the chemical, then this conformation change or conformation can be changed. That means due to the exposure of the change in heavy pH or temperature or the chemical, they again can be unfolded. So when it is unfolded to the secondary or the primary structure, then the activity again loses. So here let's say this is the normal protein. After the unfolding, now it is the inactive one. This is the inactive one and this is the active one. So the active one is called as the normal protein, or it is also called as the native protein. So when it is unfolded, then it is called as the denaturation process. The process is called as the denaturation. That means in the process the protein is unfolded to a inactive one, that is called as the denaturation of the protein. And in this protein only the primary structure of the protein remains same. That means the same amino acid is present at the denatured one and the normal one, but the secondary, tertiary and the quaternary structure is lost. Why? But the quaternary and the tertiary structure, they are maintained by these 5 structure. That means one is hydrogen bond, another is the van der Waals reaction, third one is the ionic interaction, next one is the hydrophobic interaction, and another is the disulfide bond. So these 5 factors are responsible for the formation of the tertiary and the quaternary structure. So whenever any high or the low pH or the temperature or the chemical, so then they disrupt either of the one of this factor. So when one of these factor disrupts, then it changes the conformational changes of the 3D, and once it is changed, then they again converts to the denaturation one. That means it is unfolded again to the previous one. So in the primary structure, in the denaturation process, the primary structure remains same, but in the tertiary and the quaternary structure, it has no same structure like in the native. In the secondary structure also, like in the alpha helix. In the alpha helix, in the previous slide we have studied that in the alpha helix, intra hydrogen bond is present and it maintains the alpha helix. So like that in the beta sheet. So here inter hydrogen bond is present and here it is maintained, it maintains the beta sheet. So always after the denaturation only the primary structure is present and the secondary and the tertiary and the quaternary structure are disrupted. So again the denatured protein, it can be, or it is a reversible one. That means it can be again to the native one or the normal one by the process of the renaturation. What is the renaturation? That means simply if we remove the denaturation agent, let's say in the pH, temperature, chemical, if we remove these factors, then only the denaturation protein, it can be again renaturates to form the native. That means then only it can activate or it can be functional one. So next come to what are the denaturating agents. So these are the different types of the denaturating agent. For example, a strong acid or the base, or a organic solvent, a detergent, a reducing agent, a heavy metal, heat. So they all are the denaturating agent. That means they denaturates the protein or they unfolds the protein so that the protein activity is no more. For example, what is the mechanism of the denaturation? Let's say this is the glutamic acid or this is the lysine. So here the bond is formed by the hydrogen bond. Okay, so now it is exposed to the HCl. So here H, the hydrogen, it reacts with the COO minus and it converts into the COOH, and the Cl, it reacts with the NH3 plus, and now it creates again to the NH2 plus. So by binding this, now it cannot form the hydrogen bond, because the hydrogen bond is formed, or the electrostatic bond, it can be formed by the one positive charge, one negative charge. Here the bond is formed by this positive charge, this is the negative charge, so that a hydrogen bond or the electrostatic bond can be formed. So once the HCl is released, it protonates the glutamic acid and deprotonates the lysine, so that again the lysine and the glutamate, they are separated. So it denaturates the protein by forming the protonation and the deprotonation of the amino acids. Next come to the organic solvent. For example, alcohol. So here the alcohol, simply before administration of the alcohol, here the aspartate and the tyrosine, they are joined together by the hydrogen bond. So now the ethanol, it forms the hydrogen bond to the aspartate, along with it can form the bond with the tyrosine. Here this is the alcohol, it forms the hydrogen bond with the tyrosine, it also forms the hydrogen bond with the aspartate, so that now there is no link between the tyrosine and the aspartate. So always the organic solvent, it forms the inter-hydrogen bond, so that it can disrupt intra-hydrogen bond. So always it disrupts the intra-hydrogen bond. That means the bond is formed between the aspartate and the tyrosine previously, but now after the administration of the ethanol, it forms the hydrogen bond with the aspartate, it forms hydrogen bond with the tyrosine, so it is the inter-hydrogen bond, so that it can disrupt the intra-hydrogen bond. So after that the protein can be denatured. So third one is the detergent. So detergent, the structure is like that. That means the structure is same as like that of the lipid. That means in the lipid it is called as the head part, it is called as the tail part. The head part is the hydrophilic. The tail part is called as the hydrophobic. So the detergent is called as the amphipathic. What is amphipathic? Amphipathic means any molecules which contains both hydrophilic along with hydrophobic molecules. That's why the lipid is also called as the amphipathic. That means in the head area the hydrophilic group is present, at the tail area the hydrophobic group is present. So look at the structure. This is the lipid bilayer and this is the protein. Okay, so here the interaction is called as the hydrophobic. Why it is called as the hydrophobic? Because the lipid is the hydrophobic. So here it can interact with the hydrophobic molecule of the protein. At the inside of the protein here also. So here the hydrophilic molecules. So here the hydrophilic molecules are present, because it cannot react with the or cannot interact with the hydrophobic molecules. So now the detergent is also the hydrophobic. So now it can replace the lipid bilayer by this detergent. That means before that there is the interaction between the protein to lipid. So now the bond is formed by the protein to detergent. So by that it disrupts the hydrophobic interaction between the protein and the lipid, so that it can denature. The fourth one is the reducing agent. So reducing agent, there are the many types of the reducing agent. What is the reducing agent? The agent which can reduce the molecule. For example, the urea. Another is the beta mercaptoethanol. Beta mercaptoethanol. So they are the reducing agent. So let's say, for example, this is the 2H or the H2. So that means one H, another H is present. So let's say two cysteine molecule is present. Here one it is the cysteine, here another cysteine is present, and they are found by this disulfide bond. So now one H or one hydrogen, it reduces here, another H, it reacts with this sulfur. So now one, it forms the SH, another it forms the SH. So now because of the formation of the SH or the reduction, so now there cannot be a disulfide bond is formed between that. So that's why they can be separated, or the protein folding is disrupted. So always the reducing agent that disrupts the disulfide bond formation. The beta mercaptoethanol, the urea, they are the reducing agent. So beta mercaptoethanol, it is used in the polyacrylamide gel electrophoresis. So it is used for the running of the in the gel of protein. And the protein is run in the gel. It is called the polyacrylamide gel electrophoresis. In the practical, you will know about that. So another is the heavy metal. For example, hydrogarium or the mercury. So mercury also disrupts the disulfide bond by entering between that. Here also they can disrupt the disulfide bond formation. So next one is the heat. So heat means if the temperature is increased, then there is molecular vibration. So that means the vibration will occur, and because of this vibration, the weak interaction, for example, the hydrogen bond, another is the van der Waals reaction, or van der Waals interaction. So they are disrupted by this molecular vibration. So always in the weak interaction, in the heat, weak interaction have broken down. So next come to the classification of the protein. Means the protein is divided into two parts. It is called as the simple protein, another is the conjugated protein on the basis of composition. That means, let's say this is a protein, but this protein is present in the inactive condition. It activates only when this cofactor is bound, then only it activates. So this is called as a conjugated protein. So that means the cofactor is also called as a prosthetic group. Prosthetic group. That means when this group is added to the protein, then only it activates. So before the inactivation condition, it is called as the apoprotein. When the prosthetic group or the cofactor is bound, then only it activates, then it is called as the holoprotein. And this type of the structure is called as a conjugated protein. That means it requires some cofactor, the prosthetic group. For example, the hemoglobin. In the hemoglobin, one is called as the heme and another is the globin. Globin is the protein part. Heme is the prosthetic group. Here it is the prosthetic group. Globin is the protein part and this is the apoprotein. And here the hemoglobin is holoprotein. So that means the globin is present in the inactivated condition. When the heme or the prosthetic group is bound, then only it activates and it forms the hemoglobin. So hemoglobin is the holoprotein. So after binding the prosthetic group to the apoprotein, then it forms the holoprotein. So this is the type of the conjugated protein. So what is the simple protein? Simple protein means it doesn't require any cofactor of the prosthetic group. For example, albumin, globulin, histone protein. So they are the example of the simple protein. That means it doesn't require any prosthetic group or the cofactor. So that's why this albumin is used as a standard in the protein estimation, or it is also called as the BSA, bovine serum albumin. Bovine means the cow family. The bovine serum albumin, it is a protein. It doesn't require any prosthetic group. That's why it is taken as a standard for the estimation of the protein. So now next come to another type of the protein. The protein can be divided into two parts again. One, it is called as the fibrous protein. Another is called as the globular protein. So what is the fibrous? So they are the single rod shaped structure. What is in the globular? They are the globe or the round shaped structure. So because of this rod like structure, it is insoluble to the water, while the globular are soluble to the water. So the example of the fibrous are, for example, the keratin, fibroin present in the silk fiber. So in the keratin, the secondary structure mostly it is formed by the alpha helix. In the fibroin, mostly it is present at the beta sheet in the secondary structure. That means in the fibrous structure, any structure means in the any fibrous protein like keratin, fibroin, there are other types of the fibrous protein. In the all the fibrous protein, the secondary structure are maximally made up of by single type. That means either it can form by the alpha helix or it can form by the beta sheet. But in the globular, it has the same proportion of the alpha helix and the beta sheeted. Okay. So now look at this structure. So this is the polypeptide chain of the protein. So now this is a one amino acid, this is another amino acid, this is another amino acid. So these are the structure of the amino acid. One is the alanine, one is the valine, one is the leucine. So these are the structure of the amino acid. Here one amino group is present, here CH group is present, here carboxyl group is present, here the R group is present. So all the amino acids, they are differ only by the R group. That means in the valine, the R group is this. So all these three structures are same. So in the protein, always this R group, they are protruded to the outside. So the inside, these three structure NH3CHCO-, they are present at the inside, at the main strand. Only the R group, they are outside. For example, in the alanine, let's say this is the amino acid is alanine. So here the CH3 group is found. So it is the glycine. So here the hydrogen is protruded to the outside. So always at the outside in the globular, because it is present at the soluble. It is soluble, that means it can soluble in the water. So always at the outside hydrophilic amino acid are present. At the inside, at the core, always hydrophobic, or in the green color, they are the hydrophobic amino acid are present, because at the outside obviously the aqueous solution is present, so that's why it can react or the interact with the hydrophilic molecule by forming this hydrogen bond. Here the hydrogen bond. So the amino acid, this is the, in the previous slide we have already studied that these are the hydrophilic amino acid. That means always in the amino acid or in the protein, always the asparagine, glutamine, serine, threonine, they are present in the outside, and at the inside of the core, the alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, tyrosine, they are present because they have the hydrophobic group. Here the CH3, here the CH3, so here the benzene ring. So this hydrophobic group are present. So always in, remember, it will be asked in the exam that always in the inside of the core, the hydrophobic amino acid are present, or the outside of the protein, always the polar molecule or the hydrophilic amino acid are present. So these are the all the structure of the tertiary and the quaternary structure of the protein. In the next video we will study about the myoglobin and the hemoglobin. So they are the very important protein. So next slide we will study about the myoglobin and the hemoglobin. And one more concern, many guys are watching but not subscribing it, but please subscribe my channel so that I can boost and I can bring much more videos like this. Okay.